So before I go more into the project, I just want to give you a little bit more information about this fantastic species. I'm very biased. I have been working on Hugh Billimbing for the last 20 years, and it's a wonderful animal to work with. So I'd like to share some of why it's so interesting, but also why it is a good candidate for conservation. So it has many names. So I believe that the translation for Hugh Billimbing is starfruit shark, and that is because I see leatherback shark as well, many translations. But you can see that they have these very strong ridges, very thick skin that run like the starfruit, that they have those ridges. We call it a leopard shark. So, and that is clearly that the patterns are a bit more like a leopard. However, you often see it referred to as a zebra shark, and that is because of the juvenile form. So here is a newly hatched juvenile. So these animals lay eggs, and they have the largest eggs of any shark. The babies hatch out 20 to 30 centimeters. They have these very strong banding patterns, and then they break up into these different spotting patterns, which we're not sure when they stabilize, but we can use the patterns to identify individuals. And in Australia, we now have several animals that we have tracked in the wild from patterns for up to 20 years. So we can use them for that. They do reach around 2.5 meters in total length. You will see there's one reference to 3.5 meters. I've never seen a shark that big. So I think that's an anomaly. So 2.5 is about where they get. They mature around 7 years of age. We don't know how long they live, but in the wild, the oldest animal we know of is at least 30 years old. And in an aquarium, the oldest animal is at least 40 years old. So they're fairly long-lived. So they could be much longer than that. Their teeth are small. They have sharp needle-type teeth with a plate. So they eat things like mollusks, crustaceans, and small fish as well. So one of the other things about them is that they are unique. They are the only species in their family, the Stegostomatidae. So we need to look at the order of the carpet sharks to see who are the next relatives. And these are some of the other families. These are the relatives. So you can see here the whale shark, largest fish in the sea, much bigger than Stegostoma, but they do share some features. They have similar spotting patterns, but also the whale shark has those ridges along it. Once upon a time, they were considered the same family, but they have been separated because they are quite different. Other members of that family, or the order I should say, include the wobbegong sharks, the tawny nose sharks, and also the calabaea and the bamboo sharks. So these are all in the same group of the carpet sharks. But the critical thing from a conservation perspective is that there's no other species quite like leopard sharks. So once they are gone, there is nothing there that is going to replace them. Okay, they're evolutionary, very unique. So one of the things that you could see was that really long tail. Okay, that's also a very unique feature for leopard sharks. It's the second longest tail to body ratio of any shark after the thresher shark. But they probably use it for something very different. And what we think, we don't know, what we think the purpose of the tail is that in the juvenile form, it actually seems to help them to look like a sea snake. And they swim on the surface a lot, and they swim like this. So we think that perhaps as juveniles, they are mimicking sea snakes for protection, so that they are not have greater survival. And in fact, colleagues of mine that are out hunting sea snakes for research at nighttime accidentally catch baby leopard sharks. So it's quite amazing, amazing similarity. Because they have one lobe, that's typical of sharks that sit on the bottom. However, this species swims on the surface a lot. And in Australia, where I study, my main study site, we see the adults swimming on the surface too. So perhaps they keep that tail to help them swim on the surface when they're bigger as well. Another thing a lot of people don't know is that there's two morphs, similar to manta rays, how you have a black morph and a white morph. In the leopard sharks, we have the sort of regular yellow-brown one with spots, but there's also this sandy morph, where it looks completely pale. It's not an albino, it still has brown eyes and bits of pigmentation, but it's actually just looks almost like a tawny mershark. This is a photo of the two of them together from the Indian Ocean in Western Australia. Most of the sandy morphs are found in Western Australia. They're quite rare, but there has been a recent sighting in Raja Ampat of one of these sandy morphs as well. So they are around. I've never seen it in Eastern Australia, but it's an interesting feature for these animals too. And this is a little bit of a weird fact, but something I find interesting. This species has the most spectacular leech that I have ever seen on a shark. So often sharks have their own types of parasites in a specific leech. This looks like a sea cucumber. It's not. It's a leech that was taken from the clasper of a leopard shark. You can see one attached to the shark here. And they are, as far as leeches go, actually quite pretty. I know maybe not everybody's type of animal, but banded, spectacular leech. But I've only seen, this leech only occurs on leopard sharks. I've only seen it in Queensland. I'd be very curious to know if it's found elsewhere with the distribution too. So getting back to the STAR and the restocking or rewilding project, one of the questions that was asked at this workshop was, well, okay, we have a species that we want to protect using this approach, where are we going to do it? And Raja Ampat was proposed as the location to essentially start this project. Prof. Charlie will talk more about Raja Ampat, but I will just briefly say that it was really an ideal location because we know that it's part of the distribution of the species, that they used to be there in much bigger numbers and the species had declined dramatically over the last years. Really critically, there's an extensive MPA network and shark and ray sanctuary, so there's protection for the animals when they're released, and also lots of wonderful support at different levels through community to government. So we came up with this project, STAR project, which stands for Stegosaurus tigrinum Augmentation and Recovery. It's a lot of words, much better as an acronym. And the vision statement was to re-establish a healthy, genetically diverse and self-sustaining population of Stegosaurus tigrinum. And there were some major research questions which we needed to address at the beginning. Next, please. So, is it needed? That was a really important question. So, in Raja Ampat, there's a marine protected area network, it's a shark and ray sanctuary. If we just leave it alone, won't they recover? How do we know that this is necessary and helpful? So to answer this question, we again worked with the IUCN, this time the Conservation Strategy Planning Group, who do a lot of work in this space. And we undertook a population viability analysis. So this is a way we can model into the future. We need to have the information about our species and about our location, and model into the future what is the likely recovery of the species under different scenarios. So firstly, down here, this is our business as usual. Let's not do anything. What's going to happen? It might recover. It's going to take a very long time. This is 50 years. It hasn't really passed 50 on this graph. Okay, these are modeling into the future. If there's any sort of major events, a lot of fishing comes in at some point, it will have a big impact. And there's also extinction risk. So business as usual doesn't seem like a particularly good approach going forward. Then what we can do is say, okay, what if we release animals into the wild? How will it behave then? What will happen? And we can model different scenarios. So this is a really powerful tool because it allows us to not just say, yes, restocking is helpful compared to business as usual, but we can actually use it to say, how much should we do? And also information of how frequently, how often should we be bringing in eggs? Should we release them all at once, over multiple years? And so we actually are able to use this tool to inform our approach for how we go about releasing the eggs. And so we're working under this best guess scenario, which is sort of our moderate in between, you know, the most amazing outcome, but also something that will help with the population. And so what we can see is that, okay, how many eggs should we be shipping each year? We know that we will have some mortality. Some of those eggs will not hatch, some of the babies. So how many hatchlings do we expect to come from the eggs? We also know that naturally there's mortality of some of those juveniles, so not all of those juveniles will make it into the wild. So we can model how many we expect to be releasing. And this is fantastic because this actually really helps us to set up and plan for the project. And the great news is that we're very well on track, and in fact our survival rates are better than what the prediction was for the species. So we may even reach some of this a little bit earlier. So another big consideration is, where do the animals come from? So we don't want to cause genetic pollution, where if we're introducing animals that are too distantly genetic to that population, the target population, they may overwhelm any local genetic signature, or they may also not do very well in that population if they're not adapted. And so we use population genetic information to inform this. For Hugh Billimbing, this is work that I did as part of my PhD, where we examined the population distribution for the species with 2 genetic markers. This is for the genetics group over there, the microsatellite and mitochondrial DNA, and we have a strong pattern from both types of markers that support a split through Indonesia into 2 major subpopulations. This split follows that Indonesian through-flow current, or the important biogeographic barrier of the Wallace line. And so given that Raja Ampat is over here, we are sourcing animals from this eastern population for the Raja Ampat project. So we have a team of our genetics team in the research group. We screen all of the potential breeders in aquariums around the world, and we assess which population they originate from. And it's pretty clean. Usually they fall out clearly that they come from the eastern side or the western side. In aquariums, sometimes we have mixes, so at the moment we're trying to focus on making sure that we have the animals that just come from that appropriate genetic stock. So other genetic consideration is the individual genetic diversity of the animals, so we want to avoid inbreeding. So often in aquariums, animals will be housed together that are related. So we want to screen to make sure that we're not housing animals that are not breeding animals that are too closely related, because the issue with that is that sharks are like us. You know, they get half of their DNA from mum and half of their DNA from dad. And at every one of their gene copies, gene regions, they'll have 2 gene copies, one from mum, one from dad. Most of the time, those copies will be different to each other. And this is good because if one of those copies has a bad mutation, then that won't be expressed in the animal as it gets bigger. So generally, the inbred animals tend to have more health issues and maybe a bit more mortality. So we can avoid this by screening for those animals and advising. Okay, great. Very quickly, next one, please. The other issue we have is parthenogenesis, so virgin birth. I can talk about, I'll just mention this very briefly, but in this case, we are finding in aquariums that there are examples where the females are having babies without any contribution from the male. which ends up with even more inbreeding, so we need to screen for this as well. Please have a look at the IUCN Shark Specialist Group Shark News magazine. We have an article in there which gives a lot of information on the project, as well as other wonderful articles. Next, please. Just want to say we are in the process of looking for a shark aquarist for the Mazul Resort, if anybody is interested in being part of this project. We also have a competition to name the latest leopard shark pup, and there are prizes, and the competition ends today, so get in that if you would like. But I do really want to finish on this slide, so I needed to rush a little bit. We have a sister project in Thailand that's happening and just being kicked off more recently to deal with the Western population. And very exciting news that just found out this morning. So this project has been inspired by what we're doing here in Indonesia. Just this morning we found out that they have just passed the law for full protection of leopard sharks in Thailand, which is very fantastic. And terima kasih, and thank you to all of our partners. I'd just like to have a shout out as well to say thank you to all of the fantastic Indonesian STAR team. Many of you are here, you make this project. Thank you, you guys rock.